Lampreys are any jawless fish of the order Petromyzontiformes, placed in the superclass Cyclostomata. The adult lamprey may be characterized by a toothed, funnel-like sucking mouth. The common name, lamprey, is probably derived from Latin lampetra, which may mean stone liquor, though the etymology is uncertain. Currently there are about 38 known extant species of lampreys. Although they are well known for boring into the flesh of other fish to suck the blood, only 18 species of lampreys are actually parasitic. The lampreys are a very ancient lineage of vertebrates, though their exact relationship to hagfishes and jawed vertebrates is still a matter of dispute. Characteristics. Adults physically resemble eels, in that they have no scales, and can range from 13 to 100 centimeters long. Lacking paired fins, adult lampreys have large eyes, one nostril on the top of the head, and 7 gill pores on each side of the head. The unique morphological characteristics of lampreys, such as the cartilaginous skeleton, suggest they are the sister taxon of all living jawed vertebrates, and are usually considered the most basal group of the vertebrata. Instead of true vertebrae, they have a series of cartilaginous structures called arqualia arranged above the notochord. Parasitic lampreys feed on prey as adults by attaching the mouthports to the target animal's body, then using the teeth to cut through surface tissues until they reach blood and body fluid. Although attacks on humans do occur, they will generally not attack humans unless starved. Non-parasitic lampreys, which are usually freshwater species, do not feed as adults. They live off reserves acquired as amacoetes, which they obtain through filter feeding. Hagfish, which resemble lampreys, have traditionally been considered the sister taxon of the true vertebrates, but DNA evidence suggests that they are in fact the sister taxon of lampreys. The pharynx is subdivided, the ventral part forming a respiratory tube that is isolated from the mouth by a valve called the velum. This is an adaptation to how the adults feed, by preventing the prey's body fluids from escaping through the gills or interfering with gas exchange, which takes place by pumping water in and out of the gill pouches instead of taking it in through the mouth. Near the gills are the eyes, which are poorly developed and buried under skin in the larvae. The eyes complete their development during metamorphosis, and in adults are covered by a thin and transparent layer of skin that becomes opaque in preservatives. Lampreys provide valuable insight into adaptive immune systems, as they possess a convergently evolved adaptive immunity with cells that function like the T cells and B cells seen in higher vertebrates. Lamprey leukocytes express surface variable lymphocyte receptors generated from somatic recombination of leucine-rich repeat gene segments in a recombination activating gene-independent manner. Northern lampreys have the highest number of chromosomes among vertebrates. Pouched lamprey larvae also have a very high tolerance for free iron in their bodies, and have well-developed biochemical systems for detoxification of the large quantities of these metal ions. Research. The lamprey has been extensively studied because its relatively simple brain is thought in many respects to reflect the brain structure of early vertebrates ancestors. Beginning in the 1970s, Sten Grillner and his colleagues at the Karolinska Institute in Stockholm have used the lamprey as a model system to work out the fundamental principles of motor control in vertebrates, starting in the spinal cord and working toward the brain. In a series of studies, they found neural circuits within the spinal cord are capable of generating the rhythmic motor patterns that underlie swimming. These circuits are controlled by specific locomotor areas in the brainstem and midbrain, and these areas, in turn, are controlled by higher brain structures, including the basal ganglia and tectum. In a study of the Lamprey tectum published in 2007, they found electrical stimulation could elicit eye movements, lateral bending movements, or swimming activity, and the type, amplitude, and direction of movement varied as a function of the location within the tectum that was stimulated. These findings were interpreted as consistent with the idea that the tectum generates goal-directed locomotion in the lamprey as it does in other species. Lampreys are used as a model organism in biomedical research, where the large reticulospinal axons are used to investigate synaptic transmission. The axons of lamprey are particularly large and allow for microinjection of substances for experimental manipulation. Studies have shown that lampreys are amongst the most energy-efficient swimmers. The swimming movements generate low-pressure zones around the body, which pull rather than push the bodies through the water. Distribution. Lampreys live mostly in coastal and fresh waters, although some species travel significant distances in the open ocean, as evidenced by the lack of reproductive isolation between populations. Some species are found in landlocked lakes. They are found in most temperate regions except those in Africa. The larvae have a low tolerance for high water temperatures, which may explain why they are not distributed in the tropics. Lamprey distribution may be adversely affected by overfishing and pollution. In Britain, at the time of the conquest, lampreys were found as far upstream in the River Thames as Petersham. Reduction of pollution in the Thames and River Wey has led to recent sightings in London and Chester Street. Distribution may also be adversely affected by dams and other construction projects disrupting migration routes, obstructing access to spawning grounds. Conversely, the construction of artificial channels has exposed new habitats for colonization, notably in North America, where sea lampreys have become a significant introduced pest in the Great Lakes. Active control programs to control lampreys are undergoing modifications due to concerns of drinking water quality in some areas. Life cycle. Adult lampreys spawn in rivers and then die. The young larvae, amacoites, spend several years in the rivers, where they live burrowed in fine sediment, filter feeding on detritus and microorganisms. Then, amacoites undergo a metamorphosis lasting several months. Some species do not feed after metamorphosis, while others migrate to the sea or lakes, where they feed on different species of fish and even on marine mammals. Species whose adults migrate to the sea begin preying on other fish soon after metamorphosis, even as they begin swimming downstream. Taxonomy and systematics. Taxonomists place lampreys and hagfish in the subphylum Vertebrata of the phylum Chordata, which also includes the invertebrate subphyla Tunicata and the fish-like Cephalochordata. Recent molecular and morphological phylogenetic studies place lampreys and hagfish in the superclass Agnatha or Agnathostomata. The other vertebrate superclass is Gnathostomata and includes the classes Chondrichthyes, Osteichthyes, Amphibia, Reptilia, Aves, and Mammalia. Some researchers have classified lampreys as the sole surviving representatives of the Linnaean class Cephalaspidomorphae. Cephalaspidomorpha is sometimes given as a subclass of the Cephalaspidomorphae. Fossil evidence now suggests lampreys and cephalaspids acquired as shared characters by convergent evolution. As such, many newer works, such as the 4th edition of Fishes of the World, classify lampreys in a separate group called Hyperorchia Petromyzontiformes, but whether this is actually a clade is disputed. Namely, Recently, it has been proposed that the non-lamprey hyperorchia are in fact closer to the jawed vertebrates. The debate about the systematics notwithstanding, lampreys constitute of single-order Petromyzontiformes, sometimes still seen as the alternative spelling Petromyzontiformes, based on the argument that the type genus is Petromyzon and not Petromyzonta or similar. Throughout most of the 20th century, both names were used pretty much indiscriminately, even by the same author in subsequent publications. In the mid-1970s, the ICZN was called upon to fix one name or the other, and after much debate had to resolve the issue by voting. Thus, in 1980, the spelling with a T won out, and in 1981, it became official that all higher-level tags are based on Petromyzon have to start with Petromyzoned. The following taxonomy is based upon the treatment by Fishbase as of April 2012 with phylogeny compiled by Miko Haramo. Within the order are 10 living genera in 3 families. 2 of the latter are monotypic at genus level today, and in one of them a single living species is recognized. Fossil record lamprey fossils are rare because cartilage does not fossilize as readily as bone. The first fossil lampreys were originally found in early Carboniferous limestones, marine sediments in North America. Meomyzon picoensis and hardest Yellowmontanensis from the Mississippian Mason Creek Log Estate and the Bear Gulch limestone sequence. In the 22nd of June 2006 issue of Nature, Mi Manchang and colleagues reported on a fossil lamprey from the Yizhan Formation of Inner Mongolia. The new species, morphologically similar to Carboniferous and other forms, was given the name Mesomyzon menge. The exceedingly well-preserved fossil showed a well-developed sucking oral disc, a relatively long branchial apparatus showing a branchial basket, 7 gill pouches, gill arches, and even the impressions of gill filaments, and about 80 myomeres of its musculature. Unlike the North American fossils, its habitat was almost certainly freshwater. Months later, a fossil lamprey even older than the Mason Creek genera was reported from Witterberg Group Rocks near Grahamstown, in the eastern Cape of South Africa. This species, Priscomyzon rheniensis, is very similar to lampreys found today. As food, lampreys have long been used as food for humans. They were highly appreciated by ancient Romans. During the Middle Ages, they were widely eaten by the upper classes throughout Europe, especially during Lent when they were church-sanctioned food with a much meatier taste than that of most other fish. King Henry I of England is said to have died from eating a surfeit of lampreys. On the 4th of March 1953, Queen Elizabeth II's coronation pie was made by the Royal Air Force using lampreys, especially in southwestern Europe, and in the northern half in Finland. Larger lampreys are still a highly prized delicacy. Sea lamprey is the most sought species in Portugal and one of only 2 that can legally bear the commercial name lamprey, the other one being Lampetra fluviatilis, the European river lamprey, both according to port area. Overfishing has reduced the number in those parts. Lampreys are also consumed in Sweden, Finland, Russia, New Zealand, the Baltic countries, Japan, and South Korea. The mucus and serum of several lamprey species, including the Caspian lamprey, river lampreys, and sea lamprey, are known to be toxic, and require thorough cleaning before cooking and consumption. In Britain, lampreys are commonly used as bait, normally as dead bait. Northern pike, perch, and chub all can be caught on lampreys. Frozen lampreys can be bought from most bait and tackle shops. As pests, sea lampreys have become a major pest in the North American Great Lakes. It is generally believed that they gained access to the lakes via canals during the early 20th century, but this theory is controversial. They are considered an invasive species, have no natural enemies in the lakes, and prey on many species of commercial value, such as lake trout. Lampreys are now found mostly in the streams that feed the lakes, and controlled with special barriers to prevent the upstream movement of adults, or by the application of toxicants called lampricides, which are harmless to most other aquatic species. However, those programs are complicated and expensive, and do not eradicate the lampreys from the lakes, but merely keep them in check. New programs are being developed, including the use of chemically sterilized male lampreys in a method akin to the sterile insect technique. Finally, pheromones critical to lamprey migratory behavior have been isolated, their chemical structures determined, and their impact on lamprey behavior studied in the laboratory and in the wild, and active efforts are underway to chemically source and to address regulatory considerations that might allow this strategy to proceed. Control of sea lampreys in the Great Lakes is conducted by the U.S. Fish and Wildlife Service and the Canadian Department of Fisheries and Oceans, and is coordinated by the Great Lakes Fishery Commission. Lake Champlain, bordered by New York, Vermont, and Quebec, and New York's Finger Lakes are also home to high populations of sea lampreys that warrant control. Lake Champlain's Lamprey Control Program is managed by the New York State Department of Environmental Conservation, the Vermont Department of Fish and Wildlife, and the U.S. Fish and Wildlife Service. New York's Finger Lakes Sea Lamprey Control Program is managed solely by the New York State Department of Environmental Conservation. Folklore. In folklore, lampreys are called nine-eyed eels. The name is derived from the 7 external gill slits which, along with one nostril and one eye, line each side of a lamprey's head section. Likewise, the German word for lamprey is Neunauge, which means nine-eye. Species: Geotria australis Gray 1851. Maudacia lapicida. Maudacia mordax. Maudacia precox Potter 1968. Petromyzon marinus Linnaeus 1758. Ichthyomyzon Ballium. Ichthyomyzon castaneus Gerard 1858. Ichthyomyzon fossor Rayard and Amp Cummins 1916. Ichthyomyzon Gagay Hubbs and Amp Troutman 1937. Ichthyomyzon Greeley Hubbs and Amp Troutman 1937. Ichthyomyzon unicuspis Hubbs and Amp Troutman 1937. Caspiomyzon Wagneri Berg 1906. Tetrachlorodon Geminis Alvarez 1964. Tetrachlorodon spadicius. Entasphinus Follative Vladikov and Amp Cott 1976. Entasphinus Hubbsi Vladikov and Amp Cott 1976. Entasphinus lithophagus. Entasphinus macrostomus. Entasphinus minimus. Entasphinus similis Vladikov and Amp Cott 1979. Entasphinus tridentatus. Lethanteron alaskans Vladikov and Amp Cott 1978. Lethanteron appendix. Lethanteron kamchatkam. Lethanteron kessleri. Lethanteron matsubari Vladikov and Amp Cott 1978. Lethanteron ninin asika Tuniev and Amp Renault 2009. Lethanteron reusneri. Lethanteron Zanandreri, Eudermtomyzon Graecus Renault and Amp Economidis 2010. Eudermtomyzon Stanko Karamani. Eudermtomyzon Murray. Eudermtomyzon Danfordy Regan 1911. Eudermtomyzon Helenicus Vladikov al. 1982. Judentumizon Marii, Judentumizon Vladikovi, Lampetra Epiptera, Lampetra alivariensis Mateus al. 2013. Lampetra orimensis Mateus al. 2013. Lampetra azi, Lampetra fluviatilis, Lampetra lamotini, Lampetra lanceolata kux Namp. Steiner 1972. Lampetra lusitanica Mateus al. 2013. Lampetra pacifica Vladikov 1973. Lampetra planari, Lampetra Richardsoni Vladikov and Amp. Follett 1965.